Okay, so this is subtopic 7.2, transcription and gene expression. We'll be talking about transcription in a little more detail than we did with the SL students, talking about transcription, directionality of it, how nucleosomes are useful to help regulate transcription, and post-transcriptional modification of the mRNA, including splicing. We'll talk about gene expression, what genes are expressed or not, and how that's being regulated in cells. And we're looking at examples of the promoter and looking at methylation patterns, both as applications and examples of these concepts. Here are RAS targets. I can analyze gene expression and post-transcriptional modifications and how they alter the proteome, and I can use specialized vocabulary to describe cellular processes. So coming back to the central dogma, this is the main idea we're going to talk about through all of topic 7 as well as a lot of topic 2. And what a dogma is a belief that's held with absolute certainty. So this is key in a lot of religions. We have certain things like, for example, Christians, a central dogma for them would be that Jesus Christ is the Son of God. Okay? Or in Islam, that Muhammad is the prophet, this kind of stuff. It's also such a belief held in the branch of science. So the central dogma of biology, something called the molecular biology, is that DNA will then become RNA, which then becomes a protein. So this idea of those processes, that flow of information is how cells build things, is such a key part of biology. It's called the central dogma. So what are those arrows? What do they represent in the process? Take a minute, pause video, talk to a partner. Well, the first arrow from DNA to RNA is transcription, and the second arrow, RNA to protein, is translation. Which you can also see the diagram here. Why is this so important that it's been given the title of a dogma? Well, this is how all things come to be in biology, how genes are expressed, how organs are built, all those things that come from the central dogma. Oh, it's so key to biology is given this title. And it shows a little animation showing how this whole process works. It's a really great one from Cold Spring Harbor Lab, which is known for its genetics work. So take a watch here of this video.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain sneaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it sneaks out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
It has more detail than we've seen before because at each level we talk about splicing, which we'll talk about today, as well as looking at translation. So these kind of animations are really helpful to put all the flow of all the parts of the dogma together, and I highly recommend using to help you study. Okay, let's talk about translation here. So the directionality of DNA, how does that dictate the process of transcription? So here's the same, talking about transcription earlier. We have RNA polymerase here, our big purple enzyme in this image, going down from the opening here of the DNA strand being unwound, and it's transcribing the DNA into a messenger RNA. What direction is this working in? You can see it's about the nucleotides being added to the 3' end of the DNA, so the polymerase moves in one particular direction, okay? And it's the template strand is what we're going to read off of. So RNA polymerase is like DNA polymerase. It can read in the 3 to 5 direction, 3' to 5'. So process must happen in the 5' to 3' direction. So it's adding into the 3' end of the RNA. And so the coding strand is also called the template strand, okay, to then produce the messenger RNA. So you need to memorize the directionality of this. One of the things that IB has said is important for you guys to know. So about transcription factors. So we talked about non-coding regions in 7.1, and some of those are things that are what we call transcription factors. These are going to be proteins that either encourage transcription or discourage transcription, depending on their role in the cell. So we're going to focus on 2 main ones, the promoter and terminator regions. There are many other transcription factors you will learn about if you study this more in university, but for this course, the promoters and terminators are sufficient. Luckily, their names are very logical. Can you figure out what a promoter is, what a terminator is based on that clue? Well, promoters promote transcription. So promoters are DNA sequences which are going to encourage RNA polymerase or other positive transcription factors to bind at the start of a gene. This is going to encourage transcription to happen more often, more regularly. And some genes have many, many promoter sequences. Take a minute, pause the video, talk to your partner. Why do you think that would be the case? Why would it have many promoter sequences on a single gene? Well, here's the easy promoter. Here's a promoter, and you can see here we have all these transcription factors attaching here. Then transcribe the gene. The yellow one is RNA polymerase. These are promoters are going to help encourage the gene to be transcribed. Right, promoter here, terminator here, and they're going to do so. And the multiple promoters means that gene is the proteins coded by the gene are really necessary for the cell. It needs to make a lot of copies of them very quickly, and therefore it has a lot of promoters to help encourage that transcription from happening. Okay, now terminators are at the end, and they're the sequence that tells RNA polymerase when it's time to stop doing transcription. It's the end of a gene, kind of the same way that stop codons stop translation. A terminator stops transcription. Okay, why is it important to terminate transcription? Why do we have to stop it? Why can't we just keep going? Well, we don't want to waste energy. We don't want to transcribe more than we need, and we'll waste energy both with the splicing and RNA processing, as well as potentially messing up the final protein at transcription if we do one gene's amino acids plus some extra. Look at this diagram here. We can see the terminator is included in the RNA transcript, but the promoter is not. Now, this is true only in eukaryotes. It's not true in prokaryotes, and that's because we need the terminator to help us process the mRNA correctly. Okay? Now, for those of you who are here with me in the 10th grade, these diagrams should look familiar. These are reminders of positive and negative transcription factors, also called activators and repressors. And if we look here, here is our big bubble here is RNA polymerase. If all these other transcription factors are going to assemble. Now, an activator is going to bind before the gene at a certain binding site, and that's going to help the assembly of RNA polymerase, and all the different factors that help it assemble and transcribe the gene more quickly. A repressor also binds before, and it is going to block. So something is not going to allow those genes to then be expressed. No transcription happening, which means none of those genes are going to be expressed. So this example here, again, another diagram showing this. We have RNA polymerase is going to bind to the promoter, and then the gene region in the middle is going to be coded. That actually contains the information required to make the protein in question, and the terminator at the end. So RNA polymerase binds the promoter, transcribes the gene, and ends the terminator, making our messenger RNA. Okay, so here are your first set of key ideas for the lesson. Take a minute, pause the video, and write them down. Share if you have any questions for understanding. Okay, let's continue talking about mRNA processing. So once we have transcribed the gene, we're not actually done. Technically, after the gene has been transcribed, it's not really a messenger RNA yet. It's what we call a pre-mRNA, or the RNA transcript. And it has to be processed to become a fully ready-to-go messenger RNA. So there's 3 steps to this process. There is 5' capping, there is polydenylation, also known as adding the poly A tail, and splicing. So take a look at the diagram here. What do we think are the functions of each step? So here we have our RNA transcript, or our pre-mRNA. It's a DNA, pre-mRNA here. We have to add the 5' cap, a poly A tail, and splice out those introns. And that's our actual mRNA at the end. Once it had splicing has been done, capping has been done, and polydenylation has been done as well. So the functions, well, capping and tailing are both to protect the messenger RNA and help the ribosome know where to start, where to stop, where to bind onto it. Splicing is one way to control which parts of the gene are actually going to be expressed. And there's multiple ways you can splice the mRNA, but removing unnecessary parts of the gene that are kept in the intron. And I think it was editing step. So mRNA processing is really important before translation. It has to happen, and that's going to dictate the type of protein that is being produced. If we have extra DNA, changing DNA that's spliced in, that's spliced out, or if it's left in, will change the final protein. We have a whole bunch of enzymes that do this, including poly A polymerase, RNA triphosphatase, RNA guanotransferase, RNA methyltransferase. So the 5' cap, they work on all these things. The one you need to know really is the spliceosome. So spliceosomes are a whole bunch of enzymes together. You saw it in the animation, and what are called SNRPs. And they're going to work to splice the introns out. So the introns, remember, are part of the DNA that we don't need. Okay, so in this case, in this diagram here, the green is the color code for an intron, and you can see here inside the spliceosome complex, we have the SNRPs, which are small nuclear ribonucleoproteins. And they're going to bind and create a loop of the intron, so we only have the 2 exons left, and the introns between them has been removed, has been edited out of the messenger RNA. Okay, so how does this get processed? Well, we also have the idea of alternative splicing. So what does that mean? The name is pretty logical. Alternative splicing, what do you think that means to you? Well, alternative splicing is you can splice the pre-mRNA, or the RNA transcript in multiple different ways. So we have here is we have one DNA sequence, we have one primary transcript or pre-mRNA, and it's been spliced 3 different ways. One where every single exon was kept and just all the introns were removed, and that makes this protein. One where the second exon was also spliced out, as well as a 4th, and it makes this protein. One where only the second one was not included, makes this protein. So this is a really interesting thing to disprove what used to be thought of as the one gene, one protein rule. We used to think that every gene produced only one protein. We know that's not the case, that alternative splicing is just one way in which a single gene can produce multiple different proteins. And this really helps us better understand how there's so few genes in the Human Genome Project that we've sequenced, but there's so many proteins that are being made. And the proteome is much larger than the genes we have. So humans have about 26,383 protein coding genes. We have over 2 million proteins. And alternative splicing is just one way in which we get to all those different proteins. We also process proteins differently. We do quaternary structures, combining different proteins together. There's tons of things that we do to increase the number of proteins we can get from our DNA sequence. Okay, then we have to regulate genes within us part of our lesson here, and we're getting pretty close to the end. So to regulate gene expression, what we're going to think about is, first of all, what is gene expression? We did talk about this in topic 2 and in topic 1 as well. So gene expression is controlling which genes are being kind of turned on and turned off, which ones we're going to express in the cell. I need to regulate it because we want to only produce the cells that our body needs. For example, we're always making new red blood cells, but we're not making new neurons after a certain age. So we control the type of cells we're going to produce based on when we need them. So thinking back to topic 1, cell differentiation, this is how we started out from an embryo going, and from our stem cells, going from a stem cell in either an embryo or like a pluripotent stem cells from your blood cells, and making all the kinds of cells it takes to make a human. Okay, and that's regulated at the gene level. So we regulate this different ways. So here's some different ways. Promoters or repressors can regulate which genes are being expressed. Transcription factors, methylation and acetylation of histones also allow for gene expression, and environmental factors. All of these things affect which genes are being expressed. So we do have some adaptability based on our DNA within the individual lifetime that allows us to express different genes based on the needs and different kinds of cell signals. So take a look here at this diagram. This is showing prokaryotic and eukaryotic transcription control. So here we have prokaryotic on this side, eukaryotic on this side, and we're controlling the transcription of different genes. So take a minute, pause the video, talk to her, and what do you notice is different here? What's the same, what's different? Well, in the prokaryotes, it's a lot simpler. We have RNA polymerase here with promoter, we have the coding region, the operator, and for prostate, we just have our press is going to bind onto the operator, and that's a whole level of control. In eukaryotes, we're dealing with the histones here, and then we have a whole bunch more factors to help control gene expression. And even more important to notice here is the ground state, the normal position for prokaryotic genes is they're on. Most of the time, those genes are just going to always be expressed. They only get turned off sometimes. Whereas in eukaryotic, most of the time the genes are turned off and they only get turned on sometimes. Why does this make a lot of sense in terms of those different organisms? Well, eukaryotes have way more genes than prokaryotes do. And so a lot of those genes aren't going to be necessary for every single cell, every single organ part of the body, and they are going to need to have different ones. So we have to keep them turned off. We don't waste energy and make cells and proteins we don't need. So instead, we just express the genes only when needed. Whereas in prokaryotes, it's only the unicellular. There's very few genes to express, so they're all critical to that single cell functioning. Here is an excellent compare and contrast to look at. So we have albumin is a protein being produced in the liver and in the brain. And I want you to look at how it's being controlled, gene expression, both in the liver cell's nucleus and the brain cell's nucleus. So we have the same DNA in all our cells. The control elements are here, the promoter is here, the gene is here. So what are the differences here in the way it's being controlled? Pause the video, talk to a classmate, and try to figure it out. And so after you've worked on this for yourself, here are the answers. So albumin is protein produced in the liver and used for the whole body to help with fluid regulation in your bloodstream, as well as the distribution of hormones, enzymes, and vitamins. So why do we need so much more of it in the liver than we do in the brain? And we have many more factors to help promote it in the liver than the brain. And that's because this makes sense, this is very logical, simply because, because we need much more albumin in the liver because it goes from the liver to the whole bloodstream. Whereas the brain just needs albumin for fluid regulation in that one part of it, and other parts of the brain, not nearly as much albumin to be expressed at that part of the body. Another thing for regular gene expression we're going to talk about is in the nucleosomes and histones. We did talk about these already in DNA packaging, and we'll talk about more connected here. So when DNA is bound in a nucleosome. Remember, the nucleosome is this group of 8 histone proteins binding around the DNA to attach it and supercoil it. It cannot be easily transcribed. Why is that the case? Why can't it be transcribed easily? Well, it's too tightly bound, so the DNA is not accessible, right? So look at this picture here. In the top one, this little DNA sequence here, that gene that we desired, cannot be accessed. Therefore, it's inactive. It's not being expressed. Typical hosting proteins have what we call partially charged tails. They're histone tails here. They have a positive charge on them. And that binds DNA to them very firmly because, of course, DNA is a negative charge and histone's a positive charge. So together, they're attracted. It helps hold the whole thing together really well. Now, what we can do, though, to access things is we can acetylate or do acetylation, adding an acetyl group to a histone tail, which makes it looser. So it loosens the binding. Here we can see acetylation happening here. That loosens the attraction because it's, say, also negatively charged group, and that loosens the DNA binding and makes transcription easier to happen. This makes the histones more positive, tightening the binding of DNA. The opposite process of acetylation is methylation, where we make it more positive. So acetylation makes it more negative, methylation makes it more positive, and that tightens DNA's binding and makes transcription even more challenging to happen. So how could that be used to regulate gene expression? Well, if we have more methyl groups, it's going to be a lot harder to express those genes that are highly methylated. If we have more acetyl groups, it makes it easier to express those genes that are acetylated. So acetyl equals more expression, methyl equals less expression. And those groups will change over your lifetime. So for example, here we can see DNA that is methylated versus unmethylated DNA. Okay, we have methylated DNA and unmethylated DNA, and it's going to bind differently. It's going to supercoil differently. So we have here acetylation of the green ones, and red is the deacetylation. So when it's highly methylated, so the gene's going to be switched off. It's just too tightly bound. RNA polymerase cannot bind. When a gene is switched on, RNA polymerase can bind because we have when it's more open and less tightly bound, we can actually access the gene to transcribe it. So we have found some interesting things in our environment that affect these things. So epigenetics is a really interesting field that connects how heritable traits change that does not cause by mutations in DNA changing. It's changes in gene expression or mRNA processing rather than changes in the DNA sequence. This is a relatively new part of biology, and it's really interesting. Lots to learn about it, and it's growing in knowledge all the time. People who have the same genotype can express different phenotypes based on their environment. So examples, people whose mother smoked or exposed to other sources of air pollution, they often have different sources of methylation than those who are not exposed. Weightlessness has been shown to affect methylation of genes. Imprinting behavior, like baby ducklings following their mother, that's all caused by methylation and this kind of post-birth modification of the DNA. And it's a really new field, lots to learn still, but it's really interesting. We've also found that epigenetics can be generational. So a woman who smokes while pregnant is actually could be affecting not just her daughter, but her granddaughter as well, or grandchild. Because remember, when females are a fetus, they contain all the eggs, or the pre-eggs, the parental particles that will become their babies later on. So it connects both the next generation and the following generation. And this is something really interesting. We're finding that generational trauma can affect this. There's so much interesting stuff about epigenetics that's been happening in the research. All right, key ideas, last key ideas for this lesson. And then some practice questions for you guys to work on. There's these methylation analysis questions that are really good for you guys to practice your paper 2 style questions. They're not when you do for practice. Okay? And that's it. Hopefully this was helpful for you. We're 2/3 the way through topic 7, and we'll get that rest of topic 7 done pretty soon. All right, take care, guys.